Howdy, my name is Chris Carmichael, and I'm here to talk about NF-kappa B dysregulation in bone marrow stromal and endothelial cells. And I'm from the Dr. Hoffman lab. So, one area of research that is covered in the Dr. Hoffman lab is inflammation. Now, what is inflammation? Inflammation is our body's immune response to an irritant such as pathogen or damaged tissue. Signs of inflammation include warmer temperatures, swelling, and other factors due to the increased immune response in a given area. Most commonly, we can see signs of inflammation such as simply a bruise on the skin. Now, we can talk about the specifics of what occurs during a normal immune inflammatory response. Now, what normally occurs is you have a stimulus, such as an impact, which leads to inflammation. This inflammation will lead to immune cell recruitment, which then leads to the return to function. However, in a dysregulated immune environment, such as chronic inflammation, we will have the same stimulus and inflammation, but the immune cell recruitment will not occur. This means that inflammation will feed into itself in a positive feedback loop, which leads to disease. Diseases such as diabetes and cancer, as well as stroke and cardiovascular diseases due to the increase of inflammatory cytokines. Finally, this all can lead to fatigue and depression due to the fact that an individual is unable to experience their lives to the fullest. And one of the questions that we try to tackle in the Hoffman lab is what can cause this dysregulation of the immune system. Now, before we can talk about dysregulation of the immune system, we need to talk about the immune system itself. What is in the immune system? So we have all seen these types of diagrams where we have the adaptive immunity on one side and the innate on the other, but they all come rise from hematopoietic stem cells. Hematopoietic stem cells are able to differentiate into different progenitors. One type of progenitor is the lymphoid progenitor cells, which differentiate further on into T cells and B cells, which are part of the adaptive immunity. Likewise, we also have a myeloid progenitor cells, which differentiate into monocytes, such as white blood cells and blastophils. Very importantly, hematopoietic stem cells also differentiate into red blood cells and platelets. Looking further into the issue of immune system dysregulation, we know that hematopoietic stem cells differentiate about equally into red blood cells and platelets, as well as myeloid and lymphoid in a normal environment. However, when an individual receives chronic inflammation or other immune dysregulations, we commonly see that their immune cells or hematopoietic stem cells begin to differentiate into a pro-myeloid bias. And this pro-myeloid bias is what we are tracking in the Dr. Hoffman lab as a sign of dysregulated immune system. Now, the way we track this pro-myeloid bias is through a certain protein called NF-kappa B. And NF-kappa B is very important because it releases many inflammatory cytokines and increases differentiation into myeloid cells, and is actually also part of the normal immune response to increase inflammation. We care about NF-kappa B so much that we actually created an I-kappa B knockout mouse model. In this mouse model, it lacks the machinery needed to degrade NF-kappa B itself. This means that NF-kappa B is not overexpressed tonically throughout the system, but if NF-kappa B is expressed in a certain cell, then NF-kappa B will not be degraded, thus overexpressed in the cells that do. Furthermore, we also have an NF-kappa B reporter, a fluorescent fluorophore reporter, which is very important for future methodologies. So just as a recap, we know that under normal conditions of hematopoiesis, we have a balanced lineage output, but under dysregulated inflammation, we have a disbalance in myeloid output and less lymphoid cells that come out overall. Furthermore, we have our little cute I-kappa B knockout mouse model, which has a fluorescent NF-kappa B reporter, as well as NF-kappa B overexpression if NF-kappa B occurs inside of a cell. Now, the fluorescent reporter matters a lot for FACS cytometry. FACS cytometry allows us to have data in which we are able to shine lasers through individual cells and see what is inside of them in the case of our NF-kappa B reporter. Likewise, by choosing specific antibodies on the surface of the cell, we can differentiate the cells between their different lineages, such as in my case, myeloid versus lymphoid, or in the future, the types of cells I looked into. Now, let's just get to the research. Prior research in the lab found that if we took a wild-type mouse and we transfected it with I-kappa B knockout bone marrow and let the bone marrow accumulate to its environment, once we would fax the bone marrow after a set period of time, we found that the I-kappa B knockout bone marrow would actually adapt a balanced lineage output. This is very interesting because this is the dysregulated NF-kappa B overexpressing myeloid biased bone marrow. How is it expressing a balanced lineage output? So we did the experiment again with an I-kappa B knockout mouse this time and transplanting wild-type bone marrow. Again, waiting for some time, we were able to fax the cells and find that these wild-type genetically healthy cells would have dysregulated lineage output. This is kind of interesting because it means that the myeloid bias of the cells themselves isn't dependent on their own genetics, but also can be dependent on external factors. My research looks into what kind of external factor can cause this type of dysregulation. So, looking into the bone marrow, it's actually a very diverse environment. We have everything from osteoblasts that create the bone itself, stromal cells that line the insides of the tissue, endothelial cells which line arteries and veins, neurons, and a big fraction of it is actually just hematopoietic cells, because of course your hematopoietic system, whether it's immune cells or red blood cells, come from your bone marrow. In my research, I was asking, how do stromal cells and endothelial cells, which are the structural support cells, and the cells that line the arteries and veins, have an effect, or do they even express NF-kappa B in the first place? So that leads us to our next question. Can we even see NF-kappa B in stromal endothelial cells? So inside the bone marrow itself, we see that in endothelial cells and in stromal cells, we see an increase of NF-kappa B expression. One thing to note is that I'm comparing a wild type versus an NF-kappa B reporter, which is also wild type. This means that both of these mice are genetically the same. However, we can simply see that NF-kappa B is being expressed in these cell fractions. Likewise, as a byproduct of my procedure, I was able to isolate bone cells as well, in which I was able to see endothelial and stromal cells, as well as the fact that these endothelial stromal cells in the bone itself also express NF-kappa B. This is very interesting because when we talked about what about other cells, like potentially NF-kappa B just exists sonically within the bone marrow environment and all cells express it there. So now we start asking, can we see NF-kappa B in other populations? Now, as we mentioned before, the bone marrow microenvironment is very diverse. We have neurons, we have osteoblasts, we have hematopoietic stem cells, we have all these other cells. How can we be so sure that these endothelial and stromal cells that we were able to digest are truly the only ones that are expressing NF-kappa B? So that's why we looked into the non-hematopoietic, which means non-immune, non-stromal, and non-endothelial cells in our bone marrow fractions. This means that we are looking into the neurons, the osteoblasts, everybody else that is not part of the immune system, that is not part of the stromal endothelial cells, and we can see that they actually don't express NF-kappa B above baseline. This means that those stromal endothelial cells that I was able to isolate are in fact the ones that express NF-kappa B significantly for some reason. So let's quickly take a second to recap our results. First off, we were able to find endothelial and stromal cells within the bone marrow and bone itself. Not only that, but we saw that these endothelial and stromal cells actually express NF-kappa B over their baseline. And not only that, but these are the only cell types outside of hematopoietic cells themselves that express NF-kappa B. Now this will lead us to our future steps. So in our future steps, we are going to select I-kappa B knockout mice versus wild type and see how NF-kappa B expression changes within the reporters. Furthermore, if we find that these endothelial and stromal fractions are important for NF-kappa B expression within the bone marrow, we can then use Cre-Lox models where we can stop the NF-kappa B expression in stromal or endothelial cells and see if that can help change the inflammatory dysregulation. Likewise, if we per se find that endothelial cells matter for NF-kappa B expression, then we can then start honing in on the specific types of endothelial cells that are potentially causing this NF-kappa B dysregulation. For future outcomes of my research, understanding the intricacies of our immune system may help us have positive health outcomes and improve the understanding of our immune system as a whole. Likewise, this research can help extend life expectancy as we may be able to stem the development of immune dysregulation in all individuals, from people that receive chemotherapy to people within age, which will help us all throughout life. Finally, I get to acknowledgements. I would like to thank many members of the Hoffman Lab for teaching me most of the wet lab techniques I know today, as well as Dry Lab lab analysis. And most importantly, I would love to thank my mentor Jen Shia for always being supportive in my weird side quests and helping me understand what my data actually means. And finally, here's the bibliography. Thank you.